मम्मी वो वाली पीडीएफ तो नहीं लग पाएगी, मीटिंग स्टार्ट हो गई, जब तक भैया नहीं आ जाते तब तक वो नहीं लग सकती क्योंकि वो
Good morning, Srishti. How are you?
Good morning, ma'am.
Good morning, dear. So, how many chapters? Now you people are confident. How many chapters you feel are done? I'm talking keeping in mind November, beta. Not at all September.
Ma'am, ma'am, the first unit is very well prepared, but genetics is like it is prepared, but we have to revise it again and again. I used to forget things.
That means it needs revision, huh?
Yes, ma'am.
Genetics needs revision. Okay. First unit, I saw, no doubt, very nicely done. My main concern is, no doubt, November exams only. So there, I feel we still have a month or so, although September will go in exams, but at least we have the entire October beta with you. Okay. So who else is there? Do we have anyone else in the class? No, everybody's Ayush Patel. Ayush, what are your preparations, beta? Which chapters you feel are ready?
Ma'am, my first three are ready, and in fourth one, the principle of inheritance, I am having some doubts. Not
So
that much prepared.
Same case, huh? Most of you are ready with the first unit very nicely. That is what you are saying, huh? And the second thing which is coming needs to be revised. First unit, we expect a lot of MCQs because we have more first unit only beta. Moreover, there are a lot of questions possible also there. So good that you people have prepared the first unit very nicely. That is, and how are you preparing the diagrams, children? What way are you dealing with it?
Ma'am, labeling types or like functions and that one.
Yes, basically that is what I was trying to say, that focus more on labeling part only. That is more important there with MCQ point of view. That would be more important. Okay. So let me see. Let me ask and revise from the first unit a bit, though I'll be sharing a PDF with you very soon for the next unit, but never mind. Let's ask. And you can answer as a team or answer alone, whatever you feel is absolutely okay. I'm done with it. My concern is name the mode of reproduction that ensures the creation of two new variants.
Ma'am, sexual reproduction.
Very good. Okay. Try the next one, children. Anybody can speak. What is the flowering phenomena exhibited by Strobilanthes kunthiana? What is special? What is unique about their flowering?
It flowers
Neither
every
to-
12. It flowers every 12 years.
Perfect, perfect. Name any two hermaphrodite animals.
Ma'am, earthworm and cockroaches.
Yes, tapeworm can also be included. Bache, others also speak out. Okay, please speak out, Bache. Okay, a male honeybee has 16 chromosomes. Male honeybee has 16 chromosomes, but female has 32 chromosomes. Why so? Who will explain? This is again an important one.
Ma'am, male honeybees are produced
Without
without the
fertilization.
fertilization, whereas females are produced by fertilization.
Very good. Name the animals which lay eggs outside their body.
Ma'am, oviparous.
Very good. Name asexual mode of reproduction, or as it is, we don't have it in the syllabus, but never mind. Let me take up some other questions. A microscopic spore, which is motile and which uses flagella for locomotion. Name it.
Zoospore.
Perfect. It is a zoospore. Good. Very nice. Beta, try, please. Others also try out. The process of development of gametes from meiocytes. Process of development of gametes from meiocytes. Was it the what is the process?
Gametogenesis.
Yes, it is gametogenesis. The process of development of embryo from zygote.
Embryogenesis.
Okay. Children speak out. Now tell me the correct option. Gametogenesis is a dash event. Post-fertilization. Gametogenesis.
Pre-fertilization.
And embryogenesis is pre-fertilization or post-fertilization?
Post-fertilization.
Post-fertilization.
Very good. The cyclic changes in the activities of ovaries and ducts, as well as hormones during reproductive phase of non-primates. What is the
Estrous
cycle?
cycle. Estrous cycle.
Menstrual cycle.
Very good.
Estrous
Very
cycle.
good. Very good. Okay. Female animals give birth to young ones. This is ovipary or vivipary?
Vivipary.
Okay. We'll move further now. Progenies have similar genetic makeup. Is it asexual or is it sexual reproduction?
Asexual.
Asexual.
Okay. Very good. Does amoeba undergo encystation, formation of cyst? Yes or no?
Yes.
Hmm?
Yes.
And what is the mode of division? What is the mode of reproduction when it is undergoing unfavorable conditions?
Multiple division. Multiple fission.
Yes, bache. You, everybody, please speak out. That is the better way to answer. Name the type of reproduction in which single-celled organisms generally divide.
Binary fission.
Good. Sponges reproduce by?
Gemmules.
Yes, beta. Aspergillus reproduces by? Aspergillus.
Budding.
It's sporulation, beta. Aspergillus sponges, you've already told me. The answer is what? Gemule formation. And aspergillus, they reproduce by, remember this, sporulation. Okay, next. All organisms have to pass through a particular phase before they can reproduce sexually. Before they can reproduce, what is that phase called?
Juvenile phase.
Yes, very good. Now answer this question very carefully. State the ploidy of gametes if the parent body is haploid. Yes, Viche.
Ma'am, haploid.
Haploid.
Yes, beta, if the parent body is haploid, then the ploidy of gametes is also going to be haploid only. Okay, now you tell me the fate of ovules after syngamy.
And can you repeat the question?
What is the fate of ovules after syngamy?
Seeds are formed.
Yes, and ovary?
Fruits are formed.
Good, very good. Next, Bache. Bache, try, come on, speak out, speak out. I want others also to be a part of it. Please speak out, okay? What is the function of pericarp?
It is the ovary wall.
Okay. Now let's have this. Name the two lower walls of a flower.
Corolla and calyx.
Okay. Yes. Okay, beta, the next one you tell me. Are they sterile?
Maybe or maybe may not be.
Calyx and corolla. They are called essential or they are non-essential?
Non-essential. They are sterile.
Beta, don't get confused over here. They are also called accessory. You remember, we call them as accessory or sterile. Why are they called so?
Because they don't take part in reproduction.
That is why they are called sterile. And why do we call them accessory? Accessory means what? Additional, na? Can go without it. See, accessories are what? Even if it is not present, then also the work will be done. You buy a new car, you go for accessories. But without accessories also, it's not essential that all cars will have accessories. So here we call them as accessory because we know that they are non-essential walls. A flower can go without it. Outermost protective wall of floral appendages made up of calyx is called
Sepals. Sepals.
yes, very good. They are sepals. Good. Keep answering. Mm, tell me this one now. Each stamen consists of dash and dash. What is that
Anther
stalk
and
called?
filament.
Anther
Yes,
and
yes.
filament.
Very good. Okay. Anthers in typical angiosperms have how many lobes?
Bilobe.
Bilobe.
Bilobes. Each lobe has how many theca?
Dithecus. 2.
2.
Yes, beta. They have two theca. Very good. Let's move further. So that means anther is a tetragonal structure. So it consists of how many microsporangia?
4.
4.
How many microsporangia in each lobe?
4.
4. Microsporangia develop and become? What do they become?
Pollen
Theta?
sacs. Pollen sacs.
Pollen 6.
Good. So microsporangium is surrounded by 4 walls. I'll name the outer one. You give me the other ones. First is epidermis. Next 3 are?
Endothecium, middle layers, and tapetum.
Very good. Epidermis, endothecium, middle layer, and tapetum. Very, very good. Outer 3 wall layers, you know, they are protective in function. So you know about this. What is the function of tapetum?
To
It
provide
nourishes
nourishment.
the pollen grains.
Very good, very good. When the anther is young, a group of compactly arranged homogeneous cells occupy the center of each microsporangium. What are these cells called?
Sporogenous cell.
Sporogenous tissues.
Very good. Very, very good. So each cell of the sporogenous tissue is a potential dash. What?
Pollen mother cell, a microspore mother cell.
Very good. It is pollen mother cell. The process of formation of microspores from PMC through meiosis is called?
Microsporogenesis.
Very nice. Very good. Okay. A pollen grain is a partly germinated microspore representing the dash gametophyte, male or female.
Male.
Male gametophyte.
Pollen grain is haploid or diploid?
Haploid.
Haploid.
Uninucleate, binucleate or multinucleate?
Uninucleate.
Yes, beta, remember this also, it's uninucleate. Pollen grains are spherical. How much approximate average, what is the diameter?
25 to 50
Yes, beta, it is
micron
25
meters.
to 50. Very good. It has a two-layered wall. What is it called? Pollen grains, two-layered
Intine
wall.
and exine.
Exine and
Collectively,
intine.
collectively, what is it called? Both them together, the wall is called
Sporoderm. Sporoderm.
sporoderm. Perfect. Sporoderm. Together, it is sporoderm. The outer one, you just gave me the name exine. It is made up of?
Sporopollen.
Sporopollenin.
Very good. Sporopollenin. Now, let's move further. We can preserve them as what?
Pause.
Yes, bache, tell me.
Ma'am, fossils.
Yes, beta, we can preserve them as fossils. Now be careful about the next question. You just told me the inner layer is intine. Yes, Bache. The inner layer intine, it is made up of two things. Can you name the two things?
Cellulose and
Cellulose
pectin.
and pectin.
Good, very good. Mature pollen grain in angiosperms has two cells. One is a generative cell, which is the other cell.
Vegetative
Vegetative.
cell.
Good, very nice. Vegetative cell is larger or generative cell is larger?
Vegetative.
Vegetative cell.
Which one stores reserve food?
Vegetative cell.
What is the shape of generative cell?
Spindle shape.
Circular in shape.
Yes, it is spindle shaped. And what is its location? Where will you find the generative cell?
Near the intine.
In the cytoplasm of vegetative cell.
Vegetative. Very good. In the cytoplasm of the vegetative cell. Pollen grains are generally shed at what cell stage in most
Two
of the
cell
angiosperms?
Two
Very
cell.
good.
Two-celled stage.
Yes. In the remaining species, which cell divides and gives rise to two male gametes before pollen grains
Generative
are
cell.
shed?
Generative cell.
Generative cell divides. Okay, beta, remember generative cell divides. So generative cell divides by mitosis or meiosis?
Mitosis.
Mitosis. So they are shed at what cell stage?
Three cell.
Three-celled stage.
Good. Name the grass which came to India as a contaminant with imported wheat and became ubiquitous in occurrence and caused pollen allergy.
Carrot
Name
grass.
the grass. Hmm?
Carrot grass.
Yes, can I have the scientific name? Carrot grass is absolutely okay.
Parthenium.
Yes, beta, it is Parthenium. Very good. So this shows, just was testing a bit, but 100% nice. You people are quite confident in that chapter. See this. Typical Mendelian dihybrid crosses shown below. The parents differ characters you can see. Seed shape and seed color. Please observe this cross. You can very clearly see this is a dihybrid cross. The round seed shape is dominant over the wrinkle seed shape and the yellow color is dominant over green color. The genotypes of the offsprings in the F2 generation are given as labels A to P. You can see over here A, B, C, D, E. We've given them up to P. Now please, so see the figure once very nicely. Okay, these we have mentioned from A to P. Let's try the answers. This chapter needs more revision, you said. Let me show it like this to you if you want to see the figure. Read the first, second question quickly. Just decide your answers. We'll discuss the answers after 2-3 minutes. Let me know when to scroll down, children. Okay, we'll discuss the answers one by one. First question, 5 parts, then we'll move on to the second question.
Ma'am, can you please show diagram once?
Yes, beta, let me check. I'll show you beta because you need to see. Then only you'll be able to answer, but it's not fitting in the screen. But never mind. I'll scroll up and down whenever you want. See which. See the figure, get an idea, then let's discuss the answers. Let me know, children, when you want me to go down.
Ma'am, can you scroll to the questions?
Beta, you people have seen and noted down this figure?
Yes, ma'am.
Yes, ma'am.
Okay, beta, you can have a rough idea also of the figure, then we can decide the questions. Take picture. Try to attempt all of you, even if you get it wrong, it's absolutely okay.
Ma'am, what about our biology project? Like in chemistry and physics, we have gotten lab manuals in which we have to write the experiments.
Beta, lab manuals, you don't have to write your projects actually. What I'm saying is for biology, I will not make you write projects in the lab manuals. Lab manuals are already done. You did it in the school that day. They're checked also. All those children who came to school, their work is done, submitted, checked. Lab manuals are already checked. Experiments was also performed. Were you not there in the school that day, Bachi, or you were there? Were you there in the school that day when we had
Ma'am,
We
practicals?
were not called to
ma'am,
school.
actually 12th practicals have not been done yet.
Acha, 11th class was called, 12th has not been called. Then what have you written? Without performing, you people have written practicals in physics, chemistry.
Yes, ma'am.
अरे नहीं बच्चा वेट करते हैं, we still have October and hardly 2-3 practicals are there, not much is there in the syllabus. It's better first you perform and then you write. Or we'll see, like in October first week you have holidays for 5 days. What I'll do is I'll send the written work on the group, so you finish your written work in the holidays. As of now, don't waste your time, your exams are coming 1st to 5th October, you again have holidays. So I think that would be better. One of you remind me last week of September. I'll post the pages. You can write your practicals in the holidays and then we'll perform 1-2 days whenever you come to school. Basically, DNA extraction and all has to be done. And a few spotting is there. One pollen germination. The practicals are very limited. And projects also, I don't want to trouble you now. Let's make it in October. As it is, assessment has to be done in November, beta. But projects we will not write in lab manuals. You'll make it separately. You never know, it's class 12th. They may tell us to send the projects CBSE. Randomly, what they do is they send 15 names or 15 roll numbers and they ask as evidence for assessment. So I prefer to get proper projects made and keep them safely. not the lab manuals. Lab manuals are never asked. You can simply write them and finish it off. Nobody is going to ask. But projects, beta, let's make it nicely. Don't worry about all this. Finish your exams. First week of October, you have holidays. That time you can do projects. You can make your projects and you can write the practicals. Just remind me, we'll do it that time. Okay.
Okay, ma'am.
Abhi to you just study. Abhi to you just study. That is more important.
Ma'am, also, will we be getting projects in English?
Beta, in English, we have ASL. Okay. No
Okay, ma'am.
project, we have ASL in English. So that hardly takes a day or two. We'll give you a topic you need to speak. That's it. You don't have to make any projects in English. Just the ASL will be done. I hope you've given ASL in 11th. You remember what it is?
Ma'am, we haven't given in 11th.
Not given ASL in 11th.
Yes, ma'am.
11th went like this. Only pseudo 11th is what most of the teachers call 11th for this batch. Never mind, bacha. We just give you topics and you need to speak for 2 minutes. And then we have listening skills. We'll show you something on the AV screen, audio video. You have to listen. Then we'll share a worksheet with you. Whatever you have heard, on the basis of that, you need to answer that worksheet. It's an MCQ pattern. So for you people, it will not be at all difficult. Spoken English children, though, I don't think you'll have any issues in this. As of now, relax from everything September. Just study for your exams. October first week holidays. That time we'll plan the project in files.
Okay, ma'am.
But you let me know first 5 parts when they are done.
Ma'am, can you show the diagram again? Mine has mistake
Sure,
maybe.
sure, beta. Just 1 second. I'll go back. I'll just show you. It's a dihybrid cross. See the parents. We need to practice this a little more.
Ma'am, done.
All the 5 parts are done, beta?
Yes, ma'am.
Okay. Sony, should we wait for you?
Ma'am, just a minute.
Okay, beta.
Ma'am, the WhatsApp attendance is counted or while the meeting attendance is counted?
Beta, meeting attendance is counted. WhatsApp attendance doesn't make any sense. We teachers generally take a screenshot of attendance from the participants in the meeting, beta. There are many people who write present and then disappear. That doesn't make any sense.
Yes, ma'am. Some of the students have put an attendance and they haven't joined.
I feel so that this matters, but this is for all subjects. These are instructions from school only. All the teachers, we go according to we take a screenshot of those who have joined the meeting. Sony, let me know, bacha, then we'll discuss the answers.
Yes, ma'am, done.
Chalo, let's start. What proportion of the F2 progeny, be very careful now, consists of the parental genotypes? Identify the individuals too. First, let's identify the proportion, then we'll identify the individuals.
Ma'am, option D.
1/4 consist of parental genotypes.
Yes, ma'am, option B.
How? Explain. What is your answer, Sonia Bacha? You also tell me.
Ma'am, in the F1 generation, we will be getting RR and YY, round and yellow seeds, which in the F2 generation, which we do the selfing, we will get 4 of the same types. So it will be
No,
1 by
4.
4.
Tell me about this BEIM. What are these? What is the phenotype for these?
Ma'am, capital R, capital Y, small y.
Let's move on to capital R. See, we need to find out, understand the question, but you're getting a bit stuck in the question. What proportion of the F2 progeny consists of the parental genotypes? So first, let's see what are the parental genotypes. See, capital R, small r, capital Y, small y. So where do we have this combination? Tell me quickly. Capital R,
J.
which one? Tell me, name these letters. Out of these
Ma'am,
letters.
J.
Ma'am, J.
J, capital R, yes.
No, ma'am, not J. Ma'am, F.
How F?
F.
F is what? Capital Y or small y?
Ma'am, it's J only.
See, bache? What is this?
I'm capital Y.
First, let's look for these. How many of these are there in this box? Check out how many capital R, small r, and how many capital Y, small y's are there. Obviously, there are 4.
Yes, ma'am.
Ma'am, I.
Yes, if you see this combination, capital R, capital Y, small y. So we'll get 4 combinations from this. What about this? They're round yellow only.
Ma'am, 4 from this also.
4 from this also. That is what I was trying to tell you people. It's 1/8. What is it? 1/8. Little confusing they have made it. The genotypic ratio of the individual with round. Yes, Bache.
I mean, the first one, it has 2 options, 1/8, A and P and B and N.
Let's identify A and P or B and L. Chalo, let's see. See? Tell me, A or P or B and L?
Ma'am, A and P.
Why? Why did you guess this is correct? How?
Ma'am, A has capital R and capital Y, whereas P has small r and small y.
Yes, absolutely correct. A and P. Now let us check out why not B and L. Chalo, bacha. Sony, you also try. Why not B and L? Then, why is it not B and L?
Small r, small y.
Small y, small r. Sequence is changing. The genotypic ratio of the individuals with round and yellow seeds. This too, I think you can answer without there also.
Option B, 1:2:4:2.
This is absolutely correct. 1:2:4:2. The genotype of the individual K, be careful, huh? K is similar to that of individual dash. Let's go back to
B.
K. Okay, you say M.
Yes, ma'am.
Yes, Sony, what is your answer?
Option B.
This one or this one? This is also M. Please be careful. Check out.
Ma'am, B.
Which M? B or D? Should I show you? Back. Both are M's now. So tell me which one.
Ma'am, B.
This one or this one? Sony, yes?
Second one.
Yes, bache. That means you're okay with this. How many individuals in the F2 progeny show the recombinant phenotypes? F2, how many recombinants?
Ma'am, 4.
Yes, bache. Answer from you. How many recombinants?
Ma'am,
Should
4.
I do 4 or 6?
Ma'am, 4.
4 or 6? Tell me quickly. Recombinants. How do you know it is 4, bache? Tell me.
Ma'am, recombinants is the product which we have obtained in F1 generation.
As it is, you did this ratio, beta, you never got a 6 anywhere. So obviously 4. Such a form of representation of the probability of all the possible genotypes in the progeny of a cross was developed by whom?
Punit.